Right, so we're looking at the virus bacteria worksheet. So the structure of a bacterium differs from the structure of a virus. Name two structures found in bacterium not found in a virus. So there's the bacterium. There's the bacterium wall. Inside you've got a cell membrane. You've got the flagellum, which helps move the bacterium around. You do not have a nucleus, but you have DNA as a plasmid, and you've got a nucleoid, so just like a loop. You've got cytoplasm inside. So let's write this down. That is flagellum. This here is the cell wall. That there is the cell membrane. Don't just put membrane. Get into the habit of putting cell wall and cell membrane. Right, you've got plasmid. You've got nucleoid, or they call it a circular chromosome. Right, what else have you? You've got inside here, you've got cytoplasm. Cytoplasm. And you've got a ribosome. Right, there's no mitochondria. Right, and you also have on the outside it's called a it's called a slime capsule. Right, a virus on the other hand, right, you have genetic material in the middle, and then it's surrounded by, I think it'd be easier drawing it like this, so just round, circular. It's like that's a cover. So that's the genetic material, either RNA or DNA, ribonucleic acid or deoxyribonucleic acid. This is the protein coat, and you also get you've got this membrane, and it's taken from the host cell. So the cell it infects is taken from the host cell. You'd call it a membrane, I suppose. So it's nicked from the host cell. So that's the structure of a virus, and that's the structure of a bacterium. So we can say it's got a plasmid. That's a good one to put in, and just say cell wall. But you can use most of these because they're totally different structures. Right, the diagram shows a white blood cell and some pathogenic bacteria. What did we say pathogenic was? A pathogen is an organism organism that causes a disease. Right. Explain how this white blood cell helps prevent bacteria from causing disease. So this here, remember there's two types of white blood cell. Lymphocyte and a phagocyte. A lymphocyte and a phagocyte. Right, so we need to write about this here is a phagocyte. Have you noticed the nucleus is a lobed nucleus? Normally nuclei or nucleuses are circular generally, but this has got a lobed one, and it's in the process of engulfing the bacteria and killing it. So what this does is this engulfs the pathogen. It then releases digestive enzymes and kills and breaks down the pathogen. And then what it does is, how does it prevent? Okay. So what you've got is engulfs, releases digestive enzymes, and then it just kills it. That's it. You've got it. Breaks it down, digests it. Okay. Right, again, this question we're going to do in the carbon cycle, in the nitrogen cycle. Yeah, we'll leave that. That's going to come up in the cycle. Okay, so grass cell walls are made out of cellulose. Cellulose is a carbohydrate. Okay, what are the three molecules? Carbohydrates. What are the other ones? Proteins and lipids. And remember what we said about lipids. Lipids are, if they're liquid at room temperature, we call them oils. If they are solid, we call them fats. Right, and they're saying that cellulose is the cell wall, basically, is made out of cellulose. Okay. Ooh, yeah, this takes it a stage further. So bacteria living in the large intestines of rabbits digest cellulose. Rabbits eat their own feces. Okay, that's interesting. Yes, okay. To obtain this glucose. Give two differences between the structure of bacteria and plants. Right. So I've just drawn a bacterium cell, so I'm going to draw a plant cell. So you've got cell wall, and on the inside you've got cell membrane, and then you've got a vacuole, you've got a nucleus, you've got chloroplast, you've got mitochondria, you've got ribosomes, and you've got, there you go. Right, so let's label these up. So you've got cell wall. This structure is good to know. Cell surface membrane. So we're going to call it cell membrane. You've got nucleus. No. Nucleus. You've got a, okay, I was going to put permanent vacuole. You've got cytoplasm. You've got a chloroplast, and then you've got, what do we call that? We said that was mitochondria and ribosome. Right. Plant cells have a permanent vacuole and they have chloroplasts. Right, how do we test for glucose? So remember we add Benedict's solution and you heat it in a water bath, and then it goes from blue to, I like to write the word brick red. It depends on how much and type of glucose you've got or sugar you've got. Blue to brick red. Right, what else have we got? Okay. Ooh, there's a mosquito, so you've got to read this. A mosquito carrying the West Nile virus. And then pause that and read that. And then you will end up on these questions. It's caused by a virus. 3 structures from bacterium. So you have a vacuole. You have a chloroplast, and you have a nucleus. Remember, a bacterium or bacteria does not have a nucleus. Its genetic material is in a plasmid and a circular chromosome or a nucleoid. And that is a plasmid. So just one reason why scientists predict that the situation will get worse. Question 10 to 11. Temperatures, there you go. So rising global temperatures. Rising global temperatures. It would mean that there's a change in climate, which means if it gets warmer, the mosquitoes will move into the cooler areas which are now warmer, and that's where they'll breed. Right, calculate the percentage of death rate who contacted 10 to 12. Line 10, right, 1,343 deaths. So 43 out of 1,300, and turn that into percentage, and you should get 3.3%, 3.31%. Okay. Right, give 2 abiotic factors. So remember, A Biotic is non. This makes it non-living. And biotic is living. So that might be the air, the water, the minerals, the carbon dioxide, etc. Living would be, so for example, the the trees, or the frogs, or the fungi. Mentioned. Ooh, give two abiotic factors mentioned in this passage. Okay, global warming, and so that's temperature and humidity. So they mentioned temperature and they mentioned humidity. Those are non-living, abiotic. Right. Ooh, yeah. What you could do is also mention, I suppose, I don't know, rainfall, humidity. Would you class that as the same thing? Right, explain why migration of birds may increase human outbreaks. So because the birds migrate, they travel around. Birds migrate, may have the virus, and then the mosquitoes feed on the birds, passing on the virus. And then when they migrate, they pass it on to humans. So migrating birds that have got the virus, when they arrive to that area, the mosquitoes will feed off them, and then the mosquitoes will bite human beings, feed off human beings, and they will pass on the virus. Right. It's like with anything. Do you know whenever you've got non-flowing water or pools of water, stagnant water, it's a good place for mosquitoes to breed. A good place, a good place for mosquitoes to breed is in stagnant water, or stagnant water, or still water. It's not drained, it's not moving, it's not flowing. Right. Is that a yeast cell? Okay. Right. Yeast cell. Whatever. You've got the cell wall. That's that bit. Then you've got your cell membrane. That is your vacuole. That is your nucleus. And according to this is the glycogen molecule. That's like stored energy. Chitin, the cell wall is made out of chitin, so that is F. And glycogen is C. So that's not obvious. So it's good to sort of study the structure. Anaerobic, what did we say? Anaerobic, not aerobic. Aerobic is with oxygen, oxygen, and when you put an in, it's not with oxygen. They want the word equation. So what you do is what do you call it? Respiration breaks down glucose and it releases, right, when you've got aerobic you end up with glucose. It breaks down into CO2 and water and energy. Whoa. Okay, and then for glucose it doesn't release all the energy like up there, so you end up with ethanol plus some the waste product is CO2, and you'll release some energy, but the main thing is this is, does it say fermentation anywhere? Right, okay, this is used to make alcohol by fermentation. Right, let's keep going. Okay, we've got 20 minutes, so we've got left 1, 2, 3. Okay, let's try to squeeze this in. So we're trying to investigate the effects of temperature on anaerobic respiration in yeast. So you've got a layer of, have you noticed here, you've got a layer of paraffin or oil or something. That's to stop oxygen getting in, because we want anaerobic respiration. Right, so what they're saying is, so you measure the rate of anaerobic respiration by counting the number of gas bubbles per minute. Right, so when the temperature was 20, it was 3, and it goes up 37, 36, 10, 40, 14, 45, 20. As you start getting above 45, it starts going down, because this in effect kills the yeast. It denatures the enzyme and the yeast cell dies. So it looks like as this temperature is what we would call the optimum temperature for maximum activity. And then we have to plot this. So what you do is at the bottom you'd put in the temperature, and up here you'd label this bubbles per minute, rate of anabolic respiration. So rate of anaerobic respiration, rate of anaerobic respiration in bubbles per minute. Okay, so that's that done. Now we need to look at what the temperatures, 20 to 52. So if we started at, if that was 20, would it were 25, 30, 40, 50, Yep, okay, so we can move it up even more so. Although it wouldn't matter. So if I started at 20 here, that was 30, this was 40, this is 50, so 52 would be here. Okay, so at 20 it's 3. At 20 it's, right, what do the bubbles go up to? 20. So what we could do is, if we made that 5, 10, 15, 20, okay, 5, 10, 15, 20. So each one of these is a half. You've got to be careful now. So at 20 we want 3. 25 we want 5. 25 we want 5 and then 7. 5 and 7. Now it goes to 36, 10. 36 is here and we need to go to 10 there. And it's 40, 14. 40 and 14 is there. 45 and 20. 45 and 20. And then 52 and 3. Whoa. 52 and 3. So if you look, it's, so what you do is, right, plot a graph, okay, that's it. So we've got the units, we've got the scales, it's more than half, we plotted them correctly. So we could join them. And then what's happened is basically, if you look, that there would be the, well, it could change here if you did more finer readings for temperature. That is the optimum temp. And after that, the heat will denature the enzymes, so they will die or stop working as efficiently. Okay. So explain the role of, what was the role of the paraffin? Is to stop oxygen entering, to prevent aerobic respiration. Oh, let me just finish that off. Respiration. Right, explain how the student could modify his experiment to improve his result. What he could do, as with all experiments, repeat, remove anomalies, and average out. Average out. Use a thermometer to check the temperature. You could also use any one of these, so that would get you 2 marks. That, so you use the equipment and explain why you're using it. You could use make sure that the concentration of glucose is the same. Right, make sure the type of yeast is the same. Right, another important one, and this is useful for chemistry as well, is use a gas syringe to measure volume of CO2 rather than counting bubbles. Right. So explain the results. At lower temperatures, right, this is standard answer. The kinetic energy is low, so the particles move slower, so there's less successful collisions per unit time, i.e. it is below optimum temperature. Okay, above 52. With this, the heat would denature the enzyme, so the active site, the active site, what happens to the active site changes shape, so the substrate does not fit. And when that happens, there's no reactions taking place, so the yeast is killed. Okay. Right, that's that done.